All right. Well, thank you all for attending. This is, I'm really excited for the speaker because I read the paper and I was really interested in and actually really collaborating with Dr. Ben Rosen because I think it's something that plant and animal researchers can really benefit from each other. I remember specifically during my PhD, my supervisor always said, well, look what the animal researchers are doing. That's an idea of what you can do in plants. And I want to kind of continue kind of bouncing off ideas again with animal and plants to see if we can really assist each other in the research that we do. So I am excited to introduce Ben Rosen. Dr. Ben Rosen is a research scientist at the USDA Agricultural Research Service, Animal Genomics and Improvement Laboratory in Beltsville, Maryland. He earned his PhD in plant biology from the University of California, Davis, and joined ARS in 2015. Dr. Rosen's research utilizes the latest genomic technologies to generate improved genome assemblies and pangenomes for livestock species. His research aims to enhance livestock breeding programs, improve animal health, and increase agricultural productivity. Dr. Rosen, the floor is yours, and I just remind everybody to please mute yourselves so we can enjoy Dr. Rosen's talk. Thank you.
Thanks, Jackie, for the intro, and thanks you and Anna-Rita for the invite. I'm, as you mentioned, I got my PhD in plants, but did make the switch to animals, and so I'm happy to hear that. I'm always happy to hear when folks are interested in sort of joining the two domains. So let me share my screen and see if I can get the display in the right place. So I believe you now should be looking at my talk. Is that right?
Correct.
Great. All right. So yes, Ben Rosen from USDA, and I'm going to be talking today about the Ruminant Telomere-to-Telomere Consortium, which is a large project that we've put together. It's being led by Tim Smith, also at USDA in Nebraska at USMARC, Brendan Murdoch at University of Idaho, and Stephanie McKay at the University of Missouri, along with myself. And so the project really came about because of a long-standing collaboration Tim and I have had with Adam Phillippe's group at NHGRI, Adam and Sergey Korn, working on genome assemblies. Our first project together for me was working on the goat genome, which we released back in 2017. And this was sort of the first, I guess, kind of affordable reference quality genome assembly using, combining a number of different technologies. And while it's very simple and very cheap to get much better assemblies now, at the time it was kind of novel for a smallish group to be able to put together a reference quality assembly in a fairly short amount of time. Adam's group put out, if you're aware, they put out the completed human genome recently, and telomere-to-telomere, no gaps. And so, in discussions with them about, how we can bring this to livestock, we were talking about, kind of what would be interesting, right? We could easily have done this, simply for cattle or sheep or goats, which are the main livestock that my lab works on. But we thought it would be, pretty exciting if we could, broaden the project and really sort of. Excuse me? Oh, no, okay, sorry. So we thought it would be interesting to sort of see if we could broaden the scope of things and assemble across the ruminant clade and really, sort of learn a little bit about biology along the way. And so, today I'm just going to run you through why we need telomere-to-telomere genomes, what we can benefit from those, motivations for doing it within the ruminants, what's interesting about ruminants besides, their importance to livestock production, I'll tell you about how we assemble telomere-to-telomere genomes, and then finally give you a little bit of taste of what our first draft genomes are looking like. So, to start off, when the human group released their telomere-to-telomere assembly, they added about 200 megabases worth of sequence to the human genome, which, as everyone knows, has had, billions of dollars dedicated to it throughout the years. And, this sort of sequence is roughly the size of a large chromosome, right? So they added a chromosome worth of sequence to the human genome. A lot of that sequence is found in centromeres, contained in segmental duplications. There's a lot of duplicated gene families, which were sort of missing and now are found. And then finally, the rRNA arrays are something that were always very difficult to assemble. On humans, those are found on the short arms of acrocentric chromosomes, and were always essentially just completely missing. So now those have all been placed. And, that equates to roughly 8% of the human genome. And I really like this slide from Adam Philippi, where, it's talking about whether, what's the importance of that 8% of the genome. And essentially, without it, you die. You can't do anything without centromeres, rDNA arrays. And then Sergey Koren has this sort of assembly quality test, and where he says, if I synthesize the genome that I've assembled, would I get the same organism out? And so he put that question to DALL-E and asked it to generate an image of what a woolly mammoth would look like missing 8% of its genome. And this is what came out. And as you can imagine, that's a pretty generous interpretation, since you would get nowhere without that missing 8% of the genome. And so, aside from these sort of specialized regions of the genome that are, super important, but maybe not important to everyone's research needs, what is the benefit of that T2T assembly for, a lot of folks who are just aligning data to the genome from resequencing projects, et cetera? The project showed that they were able to identify hundreds of thousands of new variants in each sample, and sort of maybe more importantly, tens of thousands of fewer false variants, right? And so they're able to get, better statistics and read mappings, and then allowing for better and more accurate variant calling. They were able to increase the number of regions where they could call methylation, and then also identify more regions where they could determine accurately the chromatin status of the DNA. So those are just sort of a few of the benefits that they were able to find. Now, one of the kind of neat things that came out of the project that I thought was that they were able to identify, really novel biology, chromosome biology in humans, right? And which, is kind of amazing, given the amount of work that's gone on for humans. But previously, as I mentioned, these acrocentric chromosomes, where the rDNA arrays resided, were unrepresented in the references, and so they weren't able to really interrogate them. And it turns out that due to the way these regions sort of localize within the nucleus, forming this, the rDNA arrays form these nucleolus region here, and are all found sort of in proximity within the genome. And because of that proximity, they actually undergo recombination. And so they've identified what they called as these pseudo-homologous regions. And, so it's sort of driven by physical proximity, but also base level similarity between these rDNA arrays. And so you end up getting recombination between the ends of these unrelated chromosomes. And so sort of one of the outcomes that they identified from that is the possibility of having these Robertsonian translocations, where you get a fusion of two chromosomes. And this is actually quite important within livestock. And it's sort of a driver of infertility within cattle. And so I was very interested to hear about this. And so this is kind of some of the things that, we're hoping to pull out signals that might, be responsible for things like Robertsonian translocations within cattle. Now, they won't be driven by the same exact mechanisms, but we can apply these similar thought processes and find these signals within other genomes. So why do we want to look at sort of telomere-telomere assemblies across the ruminant clade? Well, it's just to bring everyone up to speed who may not be aware of where ruminants lie. They are, as I mentioned, cattle, sheep, and goat. Ruminants have are the sort of unique ability to within the rumen, within their four stomachs, to convert cellulosic forage into human digestible protein. And this is obviously key for nutrition around the world. The majority of or all of the ruminants, or the majority of the ones that we typically think of as domesticated, fall within the Bovidae here. But so some of the other ruminants are, you have our deer, giraffe, and then some lesser known ruminants around. But so they're sort of sister to, we have the whales, dolphins, and hippopotamus here. And then a little bit further back, you have pigs, obviously another very important agricultural species, and then camels even further back. And so our goal is to sort of focus on the ruminants, but then extend a little bit past so we can understand sort of the origins of some of the evolutionary patterns we might identify and see what some more basal clades would tell us. So, but obviously agricultural production is a major driver of what I study and a major driver of what's important about the ruminants. Another sort of very interesting and important feature of ruminants is that there are a number of domesticated species, and these species have been domesticated at sort of different in different time frames, right? So cattle, sheep, and goat all around 10,000 plus years ago were domesticated, but there are a number of species which have been domesticated more recently, including buffalo, yak. Now there are efforts to sort of semi-domesticate bison, and even there's very recent efforts within certain places to domesticate the red deer. And so here's a deer farm in New Zealand where they are sort of starting to farm deer and starting to do genomic selection and things like that within deer. And so we can look at how these different levels of domestication and different timeframes of domestication have impacted the genome. Another feature of ruminants is that they contain quite a large number of endangered species. And so we're working with folks who are interested in certain species, such as the Dama gazelle, to try and understand their genomes and generate their genomes really as a resource for the conservation efforts and understanding and evaluating genetic diversity and population stability within these, very endangered species, where here in the case of Dama gazelle, we have, only hundreds around the world. When you consider outside of this very interesting feature of a lot of these endangered ruminants is they're found in quite large numbers in private ranches in Texas, just because there's a lot of space and a lot of wealthy ranchers who are interested in owning sort of exotic animals. But that does actually provide an opportunity for us to collect samples without having to worry about disturbing wild populations. So another interesting feature of ruminants is that they do have a broad evolutionary range. So the clade goes back about 25 million years. But within the group, we have the ability to hybridize between species, right? And so some species are interfertile, including sheep and goats. So here's an image of a geep, and these are actually fairly infrequent events because sheep and goats are have diverged quite a long time ago. But there's also more recent introgressions between species, and so there's very important trying to understand. It's very important trying to understand how yak and cattle have introgressed and how those have driven the domestication of yak. So there's both wild and domestic yak, and they are sort of co-located occasionally with cattle, and so there's people have been able to identify regions of the yak genome that have been derived from cattle, and within cattle found in these regions, vice versa. So there's also a number of other species, such as the mithun, which is a domesticated version of the guile. And so this is something that has been proposed to have been derived from an ancient introgression with cattle, but that's yet to be shown. And so we're able to look at these sort of questions and see how those introgressions have impacted the genomes in these species. So another thing is that the chromosome biology, there's actually a very rich history of chromosome biology that's been done within the ruminants. And people have been really interested in these for a number of years because of species like the muntjac, where the Indian muntjac is actually has the fewest chromosomes of any mammal. The females 2n = 6, and the males have a 2n = 7. And so they have this odd arrangement of the Y chromosome fusing with parts of the X in the male, which is very interesting. But also these two muntjac species can actually have fertile offspring. And so looking at looking at how these chromosomes have sort of been whittled down or fused together to make, down to say 2n = 6, but also seeing what happens when these species have offspring, and what happens during and what those offspring look like, and how that the centromeres, which we previously had never been able to really sequence through, seeing what these look like will be sort of fascinating. Then there's the Dama gazelle, another really interesting species where they can have anywhere from 2n = 38 to 40, and the karyotype of the offspring can't be predicted from the parents. So you can have one parent with 2n = 38, the other with 2n = 40, and you just won't know what the offspring will look like. And it's a very interesting model for chromosome evolution. So ruminants also have some really interesting characteristics of their immune system. So a lot of sort of human disease medicine folks are interested in cattle antibodies, because they produce these ultra long antibodies, which actually have potential therapeutic application as drugs for HIV. But these essentially within the IgH locus of the immune system, typically you would see a very small DJ region and very large V gene region. And in cattle and some other ruminants, there's been an expansion within the DJ region and a contraction within the V region. And so rather than, and so this is all part of, I'm not an immunologist, and so I sort of hesitate to discuss this too much, but when you have VDJ recombination to make your antibodies, a lot of that is driven by, in human, the antibody diversity is driven by the diversity of the V genes. And in cattle, they have very fewer V genes, and they think that the diversity of the antibodies is actually driven by modifications within these post-translational modifications within these this ultra long D genes along for different binding. So it's kind of cool. Now within the project, we're hoping to sort of do a number of different things, and so we've collected samples from sort of equivalent developmental stage fetuses, so that we can look at across across tissues and at a similar developmental time point to look at things like chromosome structure. So using say Hi-C, so chromatin linkages here, and also looking at methylation calling within these genomes. And then we're also going to be collecting sort of matching RNA-seq, iso-seq to look at the transcriptome. Several of our species are interspecies crosses. As I mentioned, they're interfertile. And so we have a bighorn crossed with sheep, 2 deer species, and then yak, bison, and gaur all crossed with cattle. And so we'll be able to look at how those different signals, say the chromatin organization or the methylation, is impacted in these interspecies crosses. So now I'll tell you a little bit about the species that we're targeting. So I've mentioned the cattle is up here. So this is all, this large clade is all the bovidae. So it contains cattle, yak, water buffalo, and gaur up here. These are some of the ones, and in green I have the species where we do currently have assemblies, and then in orange are species that we've identified samples and have started generating at some level, just not completed data generation. But I'll also mention up here that these are some of the sort of broader artiodactyl that we're targeting, and in the case we have a pig sample that we're working on, but we're still looking for camel and whale, sort of to mention as evolutionary outgroups to the ruminants. But here, down here within the bovidae are sheep and goats. We have a muskox, which is this actually cattle-looking thing that's more closely related to sheep and goats. And then some of those, the Dama gazelle, scimitar horned oryx. I mentioned we have an Eld's deer. Here's where the muntjacs reside. And then we have a moose sample, and here's a giraffe as well. Now we want to obviously target more broadly across the ruminant clade, and so we are sort of in the process of trying to acquire samples from some of these other ruminants, and eventually the goal is to have an assembly for each genus within the ruminants, and whether or not we achieve that goal largely depend on our ability to actually procure samples, because that has been our biggest difficulty thus far. Now, as I mentioned, there's quite a number of endangered species within this group, and so we're also targeting some of these endangered species, even if we, in some cases, have already started, say with giraffe, we've already started assembling a non-endangered subspecies, but we would like to try and provide a resource for those studying the endangered species as well. And some of the ones that we have already identified or have already started sequencing are, in fact, those ones that are endangered. And then finally, just mention that we're working with Oxford Nanopore they have an effort called org.1, where they're willing to provide resources for people who are sequencing endangered species. So if you happen to be working on endangered species and are looking for resources to do your sequencing, reach out to Nanopore. They've been great. So I'll tell you now a little bit about how we assemble our genomes. I like to sort of, for folks who are not familiar with genome assembly, liken it to, putting together a jigsaw puzzle. And the problem is that, mammalian, many plant genomes, many other insect genomes, they're highly repetitive, right? And so you can think of it, trying to assemble repeats is like trying to put together these sky pieces in a jigsaw puzzle, where, if you don't have any context from, nearby features to sort of bring things together, you're really going to be at a loss. And so the reason this is a problem for genome assembly is that when those repetitive DNA sequences are longer than your sequencing reads, you're not going to know where within those repeats these sequencing reads should be placed, right? But luckily, there have been, a large number of improvements made to sequencing, particularly long read sequencing in the recent years. And so now we're able to have much longer reads, which can span a lot of these repeat fragments. And so you would imagine that now when you have your sky pieces, they're much larger. They sort of move into more unique features, and it'll be much easier to place these things. And so it's much easier to assemble your repetitive fractions of the genome when you have these longer reads. Some of the ones that everyone's familiar with, PacBio CLR, where it's sort of going out of favor these days, but you'll probably still be seeing, folks who had previously generated assemblies, with that data. Those will still be coming out in, the next couple of years probably. But this is sort of longish reads that have a high error rate. Newer PacBio data is generated, it's called hi-fi data. And essentially what you do is you take a sort of smaller fragment, and instead of like PacBio CLR reading it once, you actually circularize it and you read that fragment multiple times. And those extra passes around that single molecule give you the ability to call a consensus for each region, since the errors are random mainly random within the sequencing. So you end up being able to have a highly accurate molecule, just sequencing read, just because you read that molecule multiple times. Then there's Oxford Nanopore, which has the ability to generate reads in the megabases. I think, I don't know what the latest numbers for the longest read generated so far, but I know that they're over 3 megabases. And so, but typically you'll see your reads between 10 and 300 KB. Another sort of a neat feature of Nanopore is that they're constantly improving their base calling ability. And so this is all done at the signal level. So you can always go back to your raw data and base call it if you've been able to store that raw data. Now that raw data is quite large, and so it's a lot of people don't hold on to that. But the error rate in Nanopore data has actually come down quite significantly, just due to that improvement in the base calling. Now they also have come out with sort of a newish technology where somewhat similar to PacBio, but in terms of having the ability to read a molecule more than once, increasing the accuracy of it. But this ONT duplex is done by when so Oxford Nanopore, and nanopore, so the DNA is pulled through a pore, and the signal is read as that DNA obstructs the pore. The change in conductance across that pore is measured, which can be translated into a base signal. And so when that molecule is pulled through, occasionally this opposite strand is pulled through immediately. And so you're able to read both strands of the DNA molecule, and therefore have the ability to sort of average a signal across those two molecules to get a more accurate call on those base pairs. This is actually further, the accuracy is increased further because Nanopore errors tend to be strand context specific, right? So when you have the molecule go through in the opposite orientation, that context changes and those bases change and to the complementary bases. And they actually have shown that some complementary sequences are much easier to read and have fewer errors than vice versa. And so you get this higher accuracy read, which has no read length dependency. And so it's just a matter of whether that second molecule has second strand of DNA is followed right afterward. And so the way that so as I mentioned, PacBio CLR is sort of no longer around. And the way that we are generating T2T assemblies and how most folks do it is that they take high accuracy long reads, so either PacBio Hi-Fi or Oxford Nanopore duplex, and they combine them with ultra long reads coming from Oxford Nanopore. And right now, that's really the only source of ultra long reads. And so in the case of our assemblies, we're looking for reads at 100 kb plus to supplement our highly accurate PacBio hi-fi data, but it's also been shown that this can be done with Oxford nanopore duplex. We just happen to be doing it with PacBio hi-fi. Right now, there's two ways to do this. You can use the software Verico that came out of was developed by Sergey Korin and Miku, and they generally recommend 50x coverage of that long accurate data, so PacBio hi-fi or Oxford nanopore duplex, 50x coverage of ultra long nanopore, and then some type of data for phasing, either that being Hi-C data or Illumina data of the parents, right? So this is the way that the assembly works, is that they take those long accurate reads. What they do is actually homopolymer compress them, and this is a way of sort of further increasing the accuracy of the reads, because most of the errors within, or a lot of frequent errors within long read data are coming from these homopolymers. And so just when you're able to collapse them, you remove a big source of those errors. Excuse me. Sorry. So yeah, they take these long accurate reads, they create an assembly graph from it, and then they are able to use align the long, ultra long reads to that assembly graph to try and actually deconvolute a lot of the issues within the graph. So these tangles and whatnot, the long reads can move through them and determine the correct paths. And then once you have your graph that's been sort of resolved with ultra long reads, you're able to take the parental information and determine a parental path, maternal or paternal, through those graphs. Sorry, getting over a little cold. So you're able to actually determine the paths, and what that allows you to do is allow you to identify the correct path through the homozygous regions in the genome. And so without knowing which direction these go, you can't really join these sort of paths, and you'll end up with mixing of haplotypes across the genome and errors within your assembly. And so it's sort of the unique combination of these different types of data allow you to get very clean, nearly complete genome, complete chromosomes out of your genome assembly. So here we are on sort of the data that we've generated to date. So when we started doing this, it wasn't entirely clear what the optimal amount of long accurate data was, hi-fi data. And so this is kind of the amount of data that we've generated for our target samples so far. And you can see some of them are quite high. And then here is the nanopore data that we've generated. And one thing I want to point out is that it's been very challenging to obtain our 30. So we sort of never even tried to reach the 50x target. We're really aiming for 30x, just because, as you can see in some of these crosses, it's taken us quite a lot of sequence. So on the left, I have the amount of total data that we've generated in gigabytes. And on the right is, this is the total data coverage. So this is for this pig cross, we've generated 325x coverage just to get slightly under 30x coverage of ultra long reads. Some of them are even more difficult. It just depends on the DNA sample. So for our muskox here, we've had to generate 300x coverage just to get 12x coverage of those 100 kb plus reads. And so it can be, depending on your sample type, etc., quite challenging. The human genomics community has worked out very good protocols for using their sort of standardized cell lines that they generate for most of their resources. and they're able to get much better ultra long conversion. We just have struggled having to deal with different samples, different tissues, different species, and so some of you folks might also struggle with it a little bit, but that's kind of normal. It takes a little bit of time to optimize. So here is what our assembly graphs look like. And so this is the graph for one of our cattle crosses. This is Wagyu cattle crossed to a Charolais cattle. These are two beef breeds. Now, this is what's known as a bandage plot, and it can look quite confusing, but essentially what we have are maternal contigs, or the maternal path is here in pink/red, and the paternal origin contigs are in blue. And you can think of these strands as essentially being, in this case, entire chromosomes. And so when they come together are those sort of homozygous regions that I mentioned before, or they are regions where the assembler just can't determine the correct path through some sort of repeat structure. And so these are actually much more complicated looking than they are in reality. And so this cattle cross here is actually has, so cattle has, each haplotype has 30 chromosomes, and we're at around 28 chromosomes each, which are near telomere to telomere. And what's happening in this big tangled mess here is, another thing I should mention is that cattle chromosomes are all acrocentric. So they essentially end at the centromere. And so there's actually a debate whether they're acrocentric or telocentric, and it all depends on what type of sequence and how much sequence is actually found on the other side of the centromere. So far, we haven't really found much on the other side of the centromere, even on chromosomes where we've been able to assemble all the way through that centromere. But at any rate, these are all tangling up at the satellite repeats within the centromere on these chromosomes. And so if you ignore the sequence that's on the other side of the centromere, which appears to be very little, the rest of the chromosome is actually completely assembled. And you can see this by the contig and scaffold N50s, which are essentially approaching the limit of what you would expect for single contigs making up an entire chromosome based on the genome sizes. So some of the things that are causing assembly issues, aside from the centromeres, are those rDNA arrays. And so like humans, cattle have 5 rDNA arrays in each parental genome, so 10 total. But unlike humans, these are not found on the acrocentric arms. These are actually found on the end of the P arm, on the end of the long arm of the Q arm, the end of the long arm on cattle chromosomes. And so you can see that they are causing some tangling, but by and large, the biggest problem for us are the satellite repeats. We do have some gaps still that are found within our chromosomes, but these gaps, the gap numbers are kind of a little bit inflated. And so here's an example of a gap that we see on the cattle X chromosome. And so this maternal haplotype here in pink has 25 total gaps in the assembly. But if you look at this one tangle, this is there's actually 7 gaps that are represented within this single tangle here. So once we're able to resolve the correct way through this path, it's actually going to be drastically reduce the number of gaps that we have within our assembly. And so the number of loci that have gaps tend to be low, and a lot of them are as I mentioned, in these satellite repeats as well. So another thing that we've noticed, or a nice feature of our assemblies, is that we're actually able to get very clean assemblies of our sex chromosomes. Now, these used to be some of the most difficult regions of the genome to assemble, and it just so happens that this combination of data is very, very effective at assembling these regions. And so we've seen very clean assemblies for most of our species for the sex chromosomes. Now, as I mentioned, those the satellite repeats are really difficult, but they are also somewhat species specific, right? And so here is our sheep cross, and what you can see is that there's even more chromosomes that are involved in this satellite repeat tangle. And so, as I mentioned before, typically cattle, sheep, goats are thought of being made up of mostly acrocentric chromosomes. There are some metacentric chromosomes in sheep, but cattle and goat both have entirely acrocentric. But it doesn't appear that we have much of any sort of sequence, like the humans have these rDNA arrays that are found on the sort of short arm of the acrocentric, whereas we have found nothing but satellite sequence. So maybe that our centromeres are actually more like mice centromeres, which are considered telocentric, because you essentially have the centromere and then the telomeres right after those centromeric repeats. As I mentioned, rDNA arrays are typically not a major driver of our tangles. And so you can see here in sheep that we're able to have pretty clean, untangled arrays for the most part, with some small exceptions. And actually, our best assembly to date would be our goat assembly, where we have really almost fully resolved our rDNA arrays, with the exception of this, tangle between chromosome 4 and 5, the paternal 4 and maternal 5. And we even have one array that was assembled all the way through. There are still, so I should clarify that even though these are identified and chromosome specific, they still have gaps within them. And so we still have to resolve the exact copy number within these repeats, but we know the classes of them. And one of them, as I mentioned, here on chromosome 28 has actually assembled all the way through it. So just to tell you a little bit more about why the rDNA array gives so much of an issue, so much assembling is, so in humans, this is the structure of the rDNA array. It's made up of, contains the 45S component of the ribosome, and which is made up of this 18S and 5S segment, which is known as the large subunit, and the 28S, which is known as the small subunit. And then it has some other content. It's slightly larger in humans because of this pseudogene, which was inserted into it. So the ruminant ones so far have been more along the lines of about 40 kb, but these are found in an array potentially hundreds of copies long. And so the way that we identify them is actually, this is a component of the ribosome, and so it's highly conserved. And so the small subunit, which is just this 28S subunit here, so here I think this is in our goat assembly, the divergence is only 0.5%. There's a larger divergence within the large subunit because it has two components separated by a spacer, and so that spacer is obviously not as conserved as those two components, and ends up getting, being responsible for larger divergence. But what we're able to do is use this software package called Ribotin to make consensus models for each of our arrays. And then once we have the copy number of the array from there's some PCR strategies to identify the copy numbers of these arrays. And then once we have that, we'll be able to go in and untangle this graph based upon the known number of copies. And so, as I mentioned, there is still this tangle here between chromosomes 4 and 5. Also, ribotin only missed very low copy arrays, and so these are actually able to be identified through repeat masker, and so it wasn't really much of an issue. And also, we can use those models that were, because it's a low copy, the models are on those chromosomes, and we don't actually need the consensus because it's just a copy or two. We can identify and create that ourselves. But here I just want to point out that when I look at this tangle in the graph, it's actually, here's the maternal array, and here's the paternal array, and there's a single sequence that is sort of making these things into a jumbled mess. What I can do is I can remove that one node and then re-input this data into ribotin, and it comes out with 2 clean arrays. And so one of the sort of major stumbling blocks to our assemblies being resolved, these rDNA arrays, is almost solved. The other issue, as I mentioned, is the satellite arrays, and I mentioned also that our issues with them tend to be different on each species. And so here is our cross between a bighorn and a domestic sheep. And what you can see, the sheep was the maternal in pink, and the sheep are all a tangled mess, as you had seen previously. But the bighorn resolves quite nicely, just like goat did, with no satellite tangles. And so in some cases, we'll be able to reduce the complexity of these tangles. Right now, this tangle has half the number of components due to the other haplotype not having any tangles, and that will help us to resolve it. A similar situation can be seen with our Gower cattle cross, where we have the Gower in blue here being completely separated from cattle, and now cattle has these sort of 2 tangles rather than a single large tangle that we have to go in and resolve. And so there's different strategies we can take depending on the different species and their ability to be crossed in order to resolve these satellite tangle regions.
I'm sorry, Ben, you have another couple of minutes, maybe?
Okay, then that was it. That was my last slide.
Sorry about that.
So yeah, no worries. Anyway, just again, like to thank some of the folks, my co-organizers of the project, and then Sergey, Miko, who have both been really helpful in the assembly process. And then finally, a postdoc with Brenda Temateo, who's also been doing a lot of work on the assemblies with me. I'm happy to take any questions. Thanks.
Great. Thank you again for a wonderful presentation. I also wanted to add in just like how one of the reasons why I really look to animals is also because I went to college undergrad to be a veterinarian, so I got a double major in zoology and genetics with a minor in animal ecology, animal behavior, and animal science, and I do research on plants. So it's one of those things I always look at. The one thing I wanted to ask you in particular is, especially with also being within the USDA, we always talk about data sharing and data management. How, with all these genome assemblies, how are you managing all the data that you're getting, and then how are you going to basically disseminate it to your shareholders?
Okay, yeah. So our goal is to have all of our data up on the Vertebrate Genome Project's Genome-ARC. So I don't know if folks are familiar with that project, but it's a large effort that's sort of integrated with the Earth Biogenomes Project to some extent. But the Vertebrate Genomes Project is being led by Eric Jarvis at Rockefeller. And they have a repository for their genome assemblies, which is publicly available, and it is hosting the assemblies, the raw data, some intermediary files, those types of things. And so we have some of our data up there now. We're working to get that. Our goal is to have all of our data up there. Eventually, we'll be submitting the assemblies to NCBI and host and the data will be there as well.
Lenore?
Yeah, hi, this is, oh, sorry, sort of a random question, but I was thinking about, you're talking about like your sample collection, and I'm wondering about how the care principles sort of may apply in this case. Like, are there, indigenous, livestock species that are important, for certain communities and cultures?
So that's possible. So certainly within livestock, you have certain breeds which are important to the communities. They have distinct characteristics and uses. Regarding the I think that some of these species that I guess that might be problematic are some of the endangered ones, and we're typically working with conservation partners. They're the ones that are, so we work with the Smithsonian on those samples, and so we try and work with the communities that are engaged in these organisms. So it's not simply us trying to collect them, but we're really trying to, and particularly it's easy for us, well, not easy, but it's relatively easy for us to assemble a genome. But we don't want to just do it if no one's out there that's interested in using that genome. And so we're really trying to have the project driven, right? We want to have a broad representation of ruminants, but within that broad representation, we want folks from sort of user communities to propose what would be most useful to them to sequence and assemble.
Monica?
Hey, thanks, Ben. Can you hear me?
Yes.
Yeah, this was a neat talk and I learned a lot. Thanks for presenting for us. One of the things I was thinking about as you were just outlining the new potential for us to get these sort of very long and accurate genome assemblies with new telomere and chromatin regions is as we keep on expanding these technologies, some of the downstream pipelines that we had baked in assumptions about, in terms of, when we had Illumina-based assemblies, how big the scaffolds were, et cetera. We've had to revise our assumptions and revise those pipelines when we realize, we can actually sequence, scaffolds that are 3 gigs long and not just 512 megs based on the human genome. So do you envision this new, more complete data, any like low-hanging fruit as to what the downstream pipelines might need to be revised for annotation or mapping or repeat identification, et cetera?
I mean, I don't know about low-hanging fruit. A lot of this stuff is really complicated. And so I know folks are working on, new mappers to try and map to repetitive regions of the genome. A lot of this stuff that we're finding is repetitive regions. And so how do you, right, it's great that they're assembled, but then if you take your shorter reads or, even, long reads are short compared to the genome, right? And even short compared to some of these repeat units, like how do you get those accurately placed? And so it's a really hard question and really hard problem. And folks are, thankfully coming up with solutions all the time, which is great.
Thanks.
But actually, so, I mean, I think some of the low-hanging fruit would be, when you do have a more complete data set, right? Like going back to some of your old data sets and maybe some of those confusing signals might actually resolve themselves. For instance, like I think a lot of work that had been done on structural variation in the past with poor genome assemblies would, really be a lot easier to interpret now with a more complete assembly.
I think we have one. Wow, sorry. You have time for one more question? I just have one more general question. So when you mentioned like pan genomes in work, like how are you going to graph all these, like visually make all these genomes almost available? Because this is a constant question, within the group is like, how are we going to make genomes viewable?
Right. Yeah, so I mean, as you saw, we have quite a number of cattle that are going to be in this project, and so those are all sort of being funneled over to our bovine pangenome project, which the having the T2T assemblies incorporated into your pangenome is going to be a huge benefit, right? Because even if all the other assemblies aren't T2T, you're still going to now have sort of the scaffolding to place some of that unplaced stuff accurately, which is great. But in terms of viewing these things, it's really still, I think, an open question. And so I know NCBI is currently trying to work on some of their comparative genome viewing tools. And so I try to expand what they have currently available to the ability to display multiple genomes, because they're certainly aware that this is something that people want to be able to do, and that's not currently done very well. So again, I think this is something we're in really early days, and hopefully those solutions come out. But I know yeah, NCBI from one is working on it. UCSC, their genome browser, I'm sure that they're considering ways in which they can better display, some of the comparative stuff that they already allow, but in a very limited fashion.
Right. With that, we are at our hour. So everyone, I would like to extend a gracious thank you to Ben for a good, really wonderful presentation. And I'd love to talk to you more about how we and plants are handling pangenomes, and how we have set up a couple of viewers, because I think that's one thing that we could work on in common. I mean, there's a lot of different things that, species wide that are very different, but some of the software part is something that we could work on.
Yeah, absolutely. I'm definitely always interested in chatting. So yeah, I would love to hear more about what you've done with plants and how we could apply that to ours. It would be great.
All right. Well, thanks again for everybody. Don't forget to check the announcements in the beginning about steering committee nominations are open. We're going to be at PAG, new working groups, and have a great November, and we'll see you next month.
Right.
Thanks, Ben.
Thanks, everyone.
Thank you.
Take care.